Welcome to this week's podcast of the Smiling Stars, and this week we are going to teach you how to create a knockout of Tetrahymena. I'm Kennedy.
I'm Ari.
I'm Reagan.
And I'm Cameron. The goal of our experiment is to create a knockout that will impact the cilia motility in Tetrahymena.
Tetrahymena are used as a model organism because they are single-celled organisms that contain two functional nuclei. They possess many core processes conserved across a wide diversity of eukaryotes, including humans. These processes aren't found in other single-cell organisms. Tetrahymena are also beneficial to use because genetic analysis and genetic tools for experimental analysis have already been developed. Specifically, in gene knockout, it is easier to perform procedures on Tetrahymena compared to mammalian cells.
You may be wondering what cilia even is. It's an organelle found on eukaryotic cells that are hair-like projections off a larger cell body. They can be both motile or non-motile. In Tetrahymena, they are motile. The structure of cilia is a little bit more complex. It consists of an inner axonemal core and an outer plasma membrane. Inside the axonemal core is a 9+2 arrangement of microtubules and a central pair of singlets. An extension off of the cilium are the dining arms. The importance of the dining arms is to attach to the microtubules and move them in a walking-like fashion, which creates a ciliary movement that we can see. Accessory proteins like heavy, intermediate, and light chain aid in the attachment of motor heads to the microtubules. In our experiment, we're going to disrupt these accessory proteins to see their true impact on ciliary movement.
Cilia movement has two phases, including a power stroke and a recovery stroke, which is what we are showing in this clip. The strokes are caused by the bending of ciliary arms, which is what Cameron explained before. Our gene of interest is the gene LC7A. It is a member of the Roadblock family of light chains and can be found both cytoplasmic and axonomal dinings. We are hoping to create a knockout of this gene, which would then affect the dining's arm ability to bind to the microtubules, therefore, hopefully in our experiment, affecting the motility of the cilia. Our first step in the experiment is DNA isolation. As a disclaimer, make sure not to contaminate this step, otherwise your procedure can't be repeated. Also, when in doubt, ask your instructor for help because they can usually tell when you're confused. In DNA isolation, we isolate genomic DNA from Tetrahymena cells in order to ensure that we have pure DNA. We have to estimate the concentration and purity of the DNA using a nanodrop. Pure DNA would be around 1.7522. Our second step is primer design. Primers are short strands of DNA that are customized and designed to fit specific nucleotides, around 20 nucleotides. Primers serve as the initial foundation for the DNA replication process. Two primers are used in PCRs and are matched to the segments of DNA. During the annealing process, primers attach to the top and bottom strands of DNA. Using the right primers is really crucial for the outcome and quality of the PCR.
Okay, so PCR, which is polymerase chain reaction, occurs in vitro, which just means that it's all occurring within a test tube. So here you can see the materials that are placed inside the test tube, and PCR basically just makes you a lot of copies of a particular fragment of DNA. You might ask, why do we even want a lot of copies? Well, it's because we want to clone these fragments into a plasmid, which we will discuss a little later. It's also really hard to identify just one fragment of DNA, so we want a lot of copies, which we call amplification, in order to run a gel electrophoresis and see the size of molecules. So we're going to start off with double-stranded DNA, and we're going to heat it up to around 95 degrees Celsius to cause denaturation. We do this in order to separate the two strands of DNA. Once separated, we cool the temperature down to around 55 degrees Celsius in order to allow those primers we selected to bind to the specific fragment of DNA, which we call primer annealing. Then we're going to heat it all back up again to 68 degrees Celsius and allow Taq polymerase to extend those primers. Why do we use Taq polymerase? Taq polymerase comes from the heat-tolerant microorganism Thermus aquaticus, and because the PCR assay occurs at such high temperatures, we use Taq polymerase specifically for its heat resistance abilities. Why do we need to heat it up anyways? Because we don't have an enzyme to separate those DNA strands, so we need heat for the denaturation. After the final extension is finished, one cycle is completed and repeat this entire process around 30 times. The polymerase chain reaction gets its name partially from the Taq polymerase used for primer extension, as well as from the chain of specific DNA fragments that is made during the thermocycling process. After each cycle, the number of DNA strands doubles. So we start off with one strand of DNA, and after one cycle, that one strand will double, and we'll have 2 strands. After 2 cycles, we'll have 4 strands, and after 3 cycles, 8 strands, and so on for the remaining cycles. This causes a chain of exponential growth in those DNA fragments. So to confirm that the PCR was successful, we are going to use a gel electrophoresis. So the gel electrophoresis simply just shows us the length of the many DNA fragments that we created. So, reminder, DNA does have a negative charge on the phosphate backbone, and because of this, when we connect the gel to a power source, the electrodes within the gel are going to create a positive and negative charge on either side of the gel. Since DNA has a negative charge, it's going to want to migrate towards the positively charged end. Separation is due to mass, so the smaller fragments of DNA will be able to travel further through the gel than the larger fragments of DNA. We will compare these results to a standardized solution of DNA, which we refer to as either the ladder or the marker, to determine the size of the DNA molecules and if PCR was successful.
Molecular cloning is a method to prepare a recombinant DNA molecule, which can replicate autonomously within a host. DNA ligation is used to physically join a DNA vector to our gene of interest, LC7A. With our pre-cut vector, we are going to insert our gene of interest. I want to note here that the vector is pre-cut for us, so by adding the gene of interest, the A's on the 3' end of LC7A will bind with the T's on the 5' end of the vector, causing hopefully perfect annealing. A couple of things to note about the vector is that it does contain an ampicillin-resistant gene, as well as already disrupted B-gal gene, which produces beta-galactosidase. So we will take the gene fragment and the open plasmid and combine them along with DNA ligase. The ends of the 2 fragments stick together by complementary base pairing. Once they are joined by ligase, the fragments become a single piece of unbroken DNA. The target gene has now been inserted into the plasmid, making a recombinant plasmid.
So the ampicillin gene that is within the vector is important because when we add our plasmids to E. coli and eventually plate them, the plates that we will use contain the antibiotic ampicillin, so only those E. coli that took in the plasmids will survive and grow colonies. If they did not take in the plasmids, they will die because they are not ampicillin resistant. We also mentioned that the B-gal gene is already disrupted, so if we are not successful in inserting LC7A, ligase will stitch together the vector without taking in the insert. This will cause the disrupted B-gal gene to become functional, and acting on its substrate X-gal, B-gal will give rise to blue colonies. But if we were successful in inserting LC7A, then ligase will stitch the insert into the vector, therefore permanently disrupting the B-gal gene. This will give rise to white colonies. However, just because the colonies are white does not mean that the insert worked, so confirmation must be performed. The confirmation of the transformation will be done using restriction enzyme digestion. So, using specific restriction enzymes, we can cut the sequence of the insert multiple times. Now, we know the total size of the plasmid with the insert, so we can predict the size of bands we expect to see on the gel electrophoresis when we cut it with the restriction enzyme. The size we see should tell us that the entire insert is in. But let's say that we have a small piece of the insert that was stitched into the vector, but the rest of the insert was not. After using the restriction enzymes, we won't see the expected pattern of bands on the gel that corresponds to the entire insert being stitched into the plasmid. This process just confirms that the transformation worked. Once we have confirmed that the transformation was successful, we're going to chop the insert with another restriction enzyme. We're then going to add a neomycin-resistant gene, which is going to cause a disruption of our gene of interest. Eventually, we will add this plasmid containing the disrupted gene to Tetrahymena, and we're going to grow the cells on neomycin plates. Just like we saw with ampicillin, if the cells took in the plasmid containing the neomycin-resistant gene, the cells will grow on the plate. This allows us to choose only those cells that contain the disrupted gene to send off.
After transformation and confirmation of gene disruption, the plasmid will be sent to a stock center. Here, it will be inserted into Tetrahymena that can then be returned to our lab for analysis. As we said in the beginning of our talk, our goal was to create a knockout that impacted cilia motility. Through phenotypic analysis, we will be able to determine if our LC7A knockout in the light chain accessory protein plays a role in ciliary motility, such as affecting the cilia beat, waveform, or how fast the tetrahymena move. You may be wondering why any of this matters. Tetrahymena is a ciliate organism whose cilia resemble those in the human body. Cilia are found in our brain ventricles, airway passages, and in females' fallopian tubes. Abnormal ciliary beating in the human body can lead to hydrocephalus. Primary ciliary dyskinesia can occur in airways, causing troubled breathing or chronic coughs. Dysfunctional or damaged cilia in the fallopian tubes can also be linked to fertility issues. Based on our findings of our experiment, we should be able to see if manipulation to the light chain accessory protein affects cilia motility, which can lead to these issues in humans. If this manipulation in fact does affect cilia motility, then through reverse genetics, researchers may be able to find ways to prevent or fix non-functional accessory proteins like the light chain, which could increase cilia motility. However, cilia is a complex organelle, and there are many proteins and factors that account for its motility, not just the one we decided to knock out. So we cannot just assume non-functional cilia or slowed down cilia are due to light chain mutations.
The overall goal of this experiment is to determine whether our gene of interest, LC7A, which is located in the light chain accessory protein and is responsible for dynein attachment and ciliary bending, plays any role in the impact of cilia motility in Tetrahymena. We use DNA isolation, primer design, polymerase chain reaction, cloning ligation, transformation, and conformation. A final transformation and conformation determine the overall success of our knockout gene. Thanks for tuning in, and don't forget to be a smiling star. Like and subscribe!